Hi, and welcome to the third video in Save Me from AP Biology. Today we're going to be finishing the review sheet for Unit 1. So in the part 1 of this video, we did basic chemistry and proteins, and now we're going to learn everything you need to know about carbs, steroids, lipids, and nucleic acids. And after that, you should be totally ready for the test. So let's get started. Notice how my box for carbs is really small. You really don't have to know a lot about carbs. Carbs, you'll know that something is a carb because it ends in ose. Glucose, fructose, sucrose, cellulose, and most of them end in ose. The term saccharide also means carb. So we call carbs that have one unit a monosaccharide. These are simple carbs. In more complex carbs, they are linked together with what is called a glycoside linkage. You will recognize carbs because they will be a ring with an oxygen in it. And they're linked together with another oxygen, so lots of oxygen going on here. Let's draw an example. Here's a ring with an oxygen in it, stuck with another oxygen onto a second ring with an oxygen in it. This carbohydrate is a disaccharide because it has two monosaccharides and one glycoside linkage. It kind of looks like without, so I'm going to change this to oxygen. There you go, ring with oxygen. So mono and disaccharides are for energy you want to use now. These are your simple sugars. Polysaccharides are more for storage. So mono or di is energy for now, easily broken down, easily put into cell respiration to make ATP. Polysaccharides are your storage molecules, and they're a little harder to break down. In fact, some polysaccharides are so hard to break down that you poop them out, and these are called fiber. So fiber is a carb. Tell that to your auntie who's low carb and see how she feels about it. The storage molecule polysaccharide in plants is called cellulose, and the one in animals is called glycogen. Notice how glycogen is a carb, but it violates the OS policy. All right, let's talk about steroids. Steroids aren't just what bodybuilders use, but I guess they are part of that. Most of the time, the words for steroids have STR or STR or something somewhere in them. It's not a hard and fast rule like with carbs, where it's always OS, or with enzymes, where it's always ACE. Steroids kind of vary, but most steroids are used as cell signals. And they're good because they can cross the plasma membrane to get inside the cell. They're hydrophobic. That's why they can cross the plasma membrane. There are lots of rings stuck together. I'm not even going to try to draw them because they're super complicated, but lots of rings. That's how you'll recognize a steroid. There are a couple of steroids you have to know. Cholesterol. See, STR. Cholesterol sticks into the plasma membrane to make the membranes more rigid. and of course testosterone and estrogen. Stir. I guess that counts as a stir. All right, that's all you have to know about steroids. You really don't have to know a lot. So let's move on to lipids. Lipid is a fancy biology word for fats. And you're going to recognize lipids because they look like zigzags. It's a whole bunch of carbons just zigzagged together. The most important thing about lipids is that they are hydrophobic. We call them fatty acids. Why do we call them fatty acids? Because it's a fat with an acid. Remember carboxylic acid? There it is. So, fatty acid. It's a zigzag with a carboxy. They are hydrophobic. Remember that means that they hate water. Some fats are amphipathic. This means that part of the molecule is hydrophobic and part of the molecule is hydrophilic. Usually the hydrophilic part is a phosphate. So they have a phosphate head that likes water. And they have a hydrophobic tail. Why don't we draw them like this? A bunch of amphipathic molecules can come together in two different ways. They can come together in a micelle. A micelle is a fancy word for bubble. We've all played with bubbles and seen bubbles in the bath. So bubbles are micelles. And basically it's a ring of amphipathic molecules where the heads are out on the outside to interact with the water and the tails go on the inside to be afraid of water. The molecules could alternatively make a lipid bilayer. This should somehow because the cell membrane is a lipid bilayer. Basically bilayer, two layers of fats. Yes, that's exactly what it is. Two layers. And they're going to look something like this. There's one layer and two layers, like a phosphate sandwich with the fatty tails being the meats. Or, cheese if you're vegetarian. All right. Sort of a common simple fat is called a triglyceride. This is made of glycerol. Glycerol looks like this. Looks like an E with O's. Plus fatty acids. 3 of them to be exact. 1, 2, 3. Fats can be saturated, which means they have no double bonds. Or they can be unsaturated, which means they do have double bonds. There are 2 types of double bonds. One's where the 2 carbons are on the same side of the double bond. That is called cis. And it looks like this. And this makes a bend in the in the fat. Trans, they're on opposite sides. Looks like this. And there's no bend. It's a nice straight fat. Why do you want a bend in your cell membrane? Well, A bend is going to make the fats more loose and fluid. You want a loose, fluid membrane if you are a creature who lives in the cold so that it can't freeze. Right? So cold creatures have cis bonds. On the other hand, if you are a warm creature, you want a saturated or trans fatty acid in your membrane so that you can pack together your membrane and you don't melt. All right, that is all you need to know about fats. We are moving on to the last section for Unit 1, and that is nucleic acids. Nucleic acids are DNA and RNA. Deoxyribonucleic acid or ribonucleic acid. You're probably going to have to know the central dogma. Sounds pretty important, right? And that is that DNA makes RNA, which makes protein, and proteins do, of course, everything. So let's start with DNA. Let's draw it. DNA, remember, is this nice double helix. There is a 5' end and a 3' end, and they run anti-parallel to each other. So this is 3', the 5' is up here. Anti-parallel just means one strand, the 3' end is at the top, and the other strand, the 3' end is at the bottom. And then they have base pairs that run along the center of this backbone. So let's talk about what that is. Anti-parallel. We already said they're running in opposite directions. There is a deoxyribose backbone. Deoxyribose. Oh, what's that? Oh, it's a sugar. And there's phosphates interspersed between the deoxyribose. The phosphates have a negative charge and are on the 5' end. And they're alternating, but the 5' end ends with a phosphate. There are two types of base pairs. The base pairs run down the center, so each of these, I guess, is a pair. You could kind of think of it more like that, with the zipper running down the center of hydrogen bonds. So there are two types of base pairs, purines and pyrimidines. I remember purines is a short word, so it's a bigger molecule, of course, because why wouldn't it be? Purines have 2 rings, and pyrimidines have 1 ring. The purines are adenine and guanine, and the pyrimidines are thymine and cytosine. A binds with T always, and G binds with C always. So you should be able to take a piece of DNA and tell me what the complementary strand is. If not now, then, eventually in a couple months when we do DNA. The zipper, I think we really actually call it a zipper, runs down the middle, and it is made of hydrogen bonds between the base pairs. Bases make a zipper. And in order to make more DNA or make RNA from DNA, you unzip the zipper. All right, how is RNA different? Well, obviously, DNA is deoxyribose, which means it doesn't have an oxygen on the ribose, and RNA is ribose, which means it has an oxygen, I guess. RNA is single-stranded, unless, you're a very specific kind of virus. And it folds from the base pairs. It has uracil instead of T. So uracil would be then a pyrimidine, and it would bond with A. There are a lot of types of RNA, but you only really have to know 3 of them. mRNA is the message in between DNA and protein. tRNA, they call transfer RNA. I like to call it translated RNA because it translates, it helps turn the mRNA into protein. Brings amino acids to mRNA to make the protein. And the last one is rRNA, which makes up ribosomes. And that is all. So let's just zoom out really quick. That is everything you need to know for Unit 1 of AP Bio on one piece of paper. Everything you need to know. If you memorize all of this, you should do pretty well on the test. If you memorize and understand all of it. I hope you've enjoyed these 2 videos, and good luck on your test.